So, joined here by Dr. Lake Paul of Bioanalysis LLC. How you doing today, Lake?
I'm doing fantastic, Brad. Thanks for having me.
Fantastic, fantastic. So you are the owner of Bioanalysis LLC, and how old is Bioanalysis?
Bioanalysis is about a year and a half old.
Okay.
Very, very new company.
Okay.
So we are being rapidly expanding and rapidly growing in the last year and a half. So,
great. And where are you located?
Philadelphia.
Philadelphia. Okay. And types of what are types of clients that you work with?
So currently we focus on gene therapy clients.
Okay.
However, we can do any type of biologics, nanoparticles, anything that is a therapeutic protein. We can analyze that using biophysics and mass spectrometry.
Okay. Okay. So what do you see as some of the trends in cell and gene therapy right now?
Oh, the trends. That's a good question, Brad. The trend right now is that the gene therapy drugs, the gene therapy medicine is a transformative medicine. So what happens is that instead of just treating the patient, you have a one-time infusion of the gene therapy product, and that basically it cures the patient of that ailment, and usually is ultra-rare diseases. So given that nature of the type of transformative medicine is, right now there's an explosion in this particular area because it can be used to treat so many diseases that have not been previously treated before. So the trend is exponential right now in terms of growth and new medicines coming out.
Sure. Now, cell and gene therapy is not necessarily new. There's been kind of a revitalization in the last few years after it was held off in the in the 80s, 90s. Tell me a little bit about that and what you've seen.
So the pioneer is Jim Wilson when he first started up in the late 90s. And unfortunately, there was a death, and that set back the field for quite a number of years now. And the Resurgence is now basically on the safety of it, which has been proven out to be pretty safe if done properly. So right now, in the last 4 or 5 years, maybe a little earlier than that, the explosion has occurred, and you see a lot of new companies coming out now. I mean, if you look at the filings, there's about 900 potential INDs, that's coming out, and then you see so much activity in terms of the programs are being developed. For example, you have, the big players like Novartis coming in, buying out of Vexus. You have, Sarepta being bought out, or buying out, and vice versa. Astellas buying out Sangamo, and so forth. I mean, there's so many interest in this field that it's just beyond explosion right now.
Sure. Well, I mean, everything seems to be, charging forward as far as the research and development and getting closer to commercialization. What are some of the roadblocks that people are hitting, and how is Bioanalysis LLC helping to break some of those down?
There are several roadblocks. One of the major roadblocks is, your packaging of your viral vectors, your capsids. And that is one of the major ones. And we address that type of roadblock in basically, we provide that service, that analytical ultracentrifugation that can quantitate your empty capsids, your partial capsids / intermediate capsids, and your full capsids. So that part of it is a critical piece, because if you know your empty capsids, you know your partial, you know your full, you can dose the client, the patient correctly. Another area that is just new, it's not even, it's not a challenge, just new, is the post-translational modification landscape of the so the AAV system or the lentivirus system, because it's such a large particle that it can, have a lot of modifications. A good example, the workhorses of the biological community is antibodies, and they usually have about 4 deamidation sites, maybe less or more. I can't remember off the top of my head. But if you look at the AAVs, you have certain systems 44 sites of deamidation, just of the asparagine, and another 58 in your glutamines. So if you look at that, it's over 100 sites to evaluate. So that just that complexity of that PTM landscape that is dependent on both the cell type, the manufacturing type, how it's processed. A great paper that came out recently by Nicole Pock really surveyed and assayed the different serotypes of manufacturing effects on this PTM landscape. So that's how we address it, is by providing an in-depth and robust service that really captures the intricacies of these particular gene therapy products.
Sure. So you were talking about some of the empty capsids and a little bit of why that's an issue. From my understanding, too many empty capsids will increase a immune response and effectively be more harmful than the drug or cure itself. Is that correct?
That is a toss-up right now between that and whether it actually promotes. So basically, you prime the system first with the empty capsids. A little bit of empty capsid might be good, might be bad. I mean, the field is, and its effect is relatively new. I call it the Wild Wild West. So it's pretty new and pretty pretty wide open right now. So a lot of these intricacies of these mechanisms is still being fully fleshed out. So, but one of the stringent things that the FDA has put in it is the percent of empty capsids. And that not only percent of empty capsid, you have to look at the percent of your partials, and then your percent of your higher order capsids, which I define as anything larger than your full capsid, AKA your aggregates. So you have to look at those type of percentages also and see what it is. Because remember, the drug is basically, the medicine is basically a one-time infusion. So there's no overall trending and tracking, and this is some really good questions. And, this is something that, someone more versed in the FDA type of specialty will be able to answer better than I would, so.
Sure. Now, AUC is one of your, just one of many, but one area of specification, for you, and sounds to be a pretty versatile tool for cell and gene therapy. In what areas, or is it fairly ubiquitous?
It's ubiquitous. So I always tell people, I'm not a one-trick pony. AUC is one of the tools in my toolbox, and besides ITC, DSC, and so forth, which I have done many experiments in that. AUC seems to be one of the most versatile tools in terms of looking at size distributions, interactions, quantitation. It's an absolute quantitative method, so you don't need standards. You just, it's not like SEC, you have to run a bunch of standards. This is, you put your sample in and you get your data, and that's absolute. So AUC is, and this has been done for many different products. I mean, I use it for nanoparticles, collagen, polysaccharides, polymers. It's all types of viruses. It is actually ideal for AAVs and viruses because, one, we have a pretty spherical shape, and two, is pretty large. Three, diffusion coefficient is very low, so it sediments rapidly, and it just is a perfect fit for the AUC. Before I turn it back to you, a little bit of history. Svedberg in 1928 did the first experiment with AUC, and it was on gold colloid particles. Think about that. Gold colloid particles in 1928. Now, come full circle, we use it, AUC, for colloids now, for polymers, but it's very versatile technique because of the optical system in it.
So why is it so difficult to get right? Why are people having issues with that right now, and how are you overcoming them?
The issues is education and knowledge. And what happened, there's a little history behind that. Back when Svedberg came out with the data with this particular technique, it exploded. Okay, it was called a flower period, and this period saw a lot of advances in the AUC field. However, the biophysics behind it, the mathematics behind it, was fairly difficult because it didn't have computers, so it became very challenging for anyone to pick it up. However, with the advent of like SDS page, mass spectrometry, sequencing, sizing column, you can get a similar type of measurement, like molecular weights and so forth, through using a very simple technique. So it went into decline. It's called an integrum phase. And what that did is that the knowledge was not transferred that well to new professors and new students and new people. So there was a gap in it until the 90s, when it really resurged, and then we had an advent of more computer-aided programs for the data fitting and so forth, that it really picked up. But still, the technique takes time. It just takes time because there's a lot of your model fitting. It's not a direct output. So to really figure out what the models are and how it fits, the curve fits, takes time. You have to see multiple samples. You have to work with multiple systems to look at it very carefully and to come up with the right fits. So that is the knowledge. And that hopefully my goal is to help transfer that knowledge. In my company, we have over 100 years of experience, not only in AUC, but everything biophysics. Wow. So it's a lot of knowledge power that we have within my company, and we do the fits right. Everything is done to a level that is not the form template. It does, a little bit of academic approach and the business approach to it too. So we do both. Okay, so that's the differentiator between my company and every other CRO out there.
Okay. So is your organization a CRO or a consultancy group?
It's both. But we do consultancy. We're aiding in consultancy a lot, but primarily what we're trying to focus on is as a service provider. We're trying to bring back that scientific robustness, that personalized attention, and that customer service that I think is lost these days. We're not going to nickel and dime you in data transfer. That's your data. If it requires us to do an extra experiment to prove out the data, we will do it at no cost, because that's the way we approach our business. The science is the business. The business is not the science. Okay, that's my motto.
Very nice. So there's a lot of research that's coming to you that's being done by individuals prior to you. So broken research or broken data. How are you overcoming some of those challenges? Is it in design? Is it in analysis? All of the above?
All of the above. We talk to the client, and our team is so diverse and so well knowledge that we can start designing experiments as you're telling us what you want. So we already during our initial conversation, we'll ask you some key questions to get some information, and at that same point we're designing the experiment that will answer those questions. There's a lot, that's how we take the broken research and try to put it together a little bit. Okay.
So are you doing GLP analysis, or also do you do GMP as well?
Great question, Brad. We are doing non-GLP/GMP. However, in February, the GLP GMP lab is opening in Philadelphia. That is going to primarily focus on release test method development using AUC. And then we're going to expand after that. But right now, there's a need for good AUC release tests. Once it's done properly, nothing touches it.
Sure. Now, AUC is a big topic. It's, something we've touched on as far as, one of your expertise. What else, what else can Bioanalysis LLC do? What else can you bring to the table?
Oh, great. Besides AUC, we do circle dichroism, isothermal titration calorimetry, SECMALS is multi-angle light scattering, dynamic light scattering. So all of the biophysics we perform those experiments. And that is something that we will bring those experiments up, depend on the project. So for example, if you're just looking at if you want to find out a binding interaction, depends on what the sample is, we might use surface plasmon resonance or ITC, or we might just do it in SECMALS or AUC. It all depends on the material, the question, and what you want to get out of it. But we do have a very large toolbox that we use. We primarily focus on AUC because it's very critical for the gene therapy market, but please bring these challenging products so we can do something else besides AUC.
So, I mean, you were the architect of this business. This was, something that that you started. Many other companies were doing some similar levels of work. What made you think that there was an opening in the market for you?
It has to be done right. It just has to be done right. And the analysis has to be done properly. So we don't apply the form template. It's a personalized approach, kind of like what you do, Brad, a personalized approach to your company. And that is the key indicator, because I have used CROs before in which they just give me a couple of pages of report and that's it. And I'm like, what is this? I have more questions. So what we do, I do the same thing. I don't do that same thing. I expand upon that, and it's that personalized care of the clients, and that attention to detail that is really sets my company apart from everyone else.
Tell me, I mean, don't go into any proprietary or NDA information, but what separates some of your reports from other data that you see out there in general?
The reports are very detailed, and we try, so the reports are very detailed, but we give it a summary section in which you can read the first 2 pages and get everything you need. But once you go in and dive deeper into the report, there's a lot of intricacies that we talk about, a lot of subtleties that we talk about in the reports, that if 5 years from now you want to figure out what we did, you can read that report and be like, oh, that's exactly what they did. And there's our approaches. We not only use the current programs that are out there, we have custom programs that we've built, and we do orthogonal analyses to solidify our data analysis. Because remember, these are all curve-fitted data. So that's for the AUC. For the mass spectrometry, we do the initial mapping and so forth by using the computer with database searches, but then we validate it. We take the time to make sure that the database searches are calling the right peptides what it is, because we find a lot of false positives. When we look at, for example, deamidation is a very tough one to figure out, and we have a workflow that is pretty robust, that when we call a site deamidated, you best know it is deamidated. Okay, that's the confidence we have in our data analysis.
Sure. So, as I said, you're the architect of this business. What surprised you the most when starting up your own shop, starting up your own business?
The gray hair. The amount of gray hair I've gotten since the last a year and a half. What surprised me that people are tired of form template type of analysis. People are just tired of it. As a scientist, you don't want just a form template. Maybe at some point, some levels, some projects require a form template and a form analysis. But most of the time, when you're in the research and process development, things are changing, things are moving. You have to be adaptable to that. We just can't apply the same analysis over and over and over to the same sample and expect to get the same result. You do, actually, let me back up a second, you do. But that is when you are going into the GMP section. That's when the same rote analysis is done over and over and over, and you get the same reproducible results. That is for when you go into method development and your GMP. However, you're going to get samples that it's unknown. So how do you apply for that? How do you figure it out? You can't just do the same thing over and over.
Right, right. Do you see in larger organizations that are doing similar data analysis that it is rote template? It is just what they're used to, and trying to break out of that paradigm. Do you see people having issues being stuck in that?
Yeah, that's the thing. They're unfortunately that's what is being done. I've consulted too many times to see the same thing over and over and over and over, when at the end of the day you can't do that. For example, I've seen projects in which it was called, we cannot analyze this, because they don't know how to do it. And I was called in and I used the same data and figured out everything for it. You just have to have a different experience and knowing what you need to do to figure out these stuff.
Sure, sure. Well, that is fantastic. Well, how do people get in contact with you? What's the best way? I don't need you to hand out your cell phone number, but what's the best way for people to reach you?
The best way for people to reach me is by my email, lpaul@bioanalysis.llc.com. I'm on LinkedIn, very active you can find me. And my name is Lake Paul, so it is a very easy searchable name. The website is being developed right now, and that should be launched in January. And we're all excited. We're growing here, and it's just absolutely fantastic.
Oh, fantastic! Is your email accessible on LinkedIn, or does somebody have to connect with you to get your email?
It's accessible on LinkedIn. I made sure of that.
Fantastic. Well, great. Well, listen, just wanted to thank you very much for coming on. It was great finding out, a little bit more about your organization. Where do you see you guys? Where do you see yourselves growing in the next few years?
Oh, well, we're planning to do a lot of the growth is basically in solidifying the GMP release using AUCs, bringing more of the assays in-house, expanding our portfolio for different clients. So, I mean, the sky is the limit right now.
Sure.
Yeah.
Well, fantastic. Best of luck with that. And listen, thanks for sitting down and chatting with me.
Thanks for the great questions, Brad. It's an absolute pleasure.
All right. Have a great one.